So good morning, all of you. Again, for another year of exam focus 2024. So today we are going to deal with an important topic, that is the development of tooth. It is one of the chapters where like a lot of questions will come, and main essay questions also are being frequently asked from this chapter. Now, when we are going for this development of tooth, there are some basic things that you have to remember, and so the parts of the teeth. You know that when you take a cross section, the histology is you have enamel, which is a hard tissue. Again, dentine is a hard tissue. You have pulp, which is a soft tissue. Then you have cementum, again a soft tissue, and alveolar bone. So these are all the components and the supporting structures of the teeth, and periodontal ligament again. So when we talk about the development, you have to understand that enamel is something that is formed from ectoderm, and dentine, pulp, cementum, periodontal ligament, and alveolar bone are all formed from ectomesenchyme. I hope all of you know what is ectomesenchyme. Ectomesenchyme is the connective tissue which is derived from neural crust cells, and so that is the reason why it is called ectomesenchyme. And the other parts of the body, the connective tissue is derived from the lateral plate mesoderm, and uh, for tooth development, the connective tissue is derived from neural crust cells, and that is why it is called ectomesenchyme. Now, like if somebody asks you what is a tooth germ, that is actually a primitive structure from where the tooth develops. That is what you call as a tooth germ. So usually the development of the tooth germ will start developing by the third week of intrauterine life, and you have to know like what are the different components of the tooth germ. These are some basic things that you have to keep in mind. So what we have, what are the different components of the tooth germ? We have enamel organ. So enamel organ is an ectodermal structure, and it is from this enamel organ enamel is being formed. Then what we have is a dental papilla. Dental papilla is an ectomesenchymal structure. It is from where dentine and pulp is being formed. And dental follicle or dental sac is again another ectomesenchymally derived structure. It is from where the cementum, periodontal ligament, and alveolar bone is also being formed. So that is what is being shown here. So easily you will be able to identify the enamel organ produces enamel, dental papilla, which is an ectomesenchymal structure, produces dentine and pulp. Dental sac produces cementum, periodontal ligament, and alveolar bone. So this is something that the basic things that you have to keep it in mind. Okay, so when we are actually talking about the tooth development, it's a pretty big, huge chapter, and it is quite impossible to complete the whole thing during this exam review program. Maybe like we have only 45 minutes or like half an hour to for one session. So I will be dealing. I'm not going into the short notes or brief notes, 5 marks or 3 marks questions. I will be focusing more on the frequently asked essay question. And so there are the two frequently asked essay questions are: define odontogenesis, enumerate various stages of odontogenesis, and discuss advanced bell stage of tooth development with the help of a neat diagram. And another question that has been frequently asked is enumerate the life cycle of ameloblast and discuss in detail amelogenesis. So these are the two questions that I am going to discuss in this particular session. So if a question is being asked about define odontogenesis and enumerate the various stages of odontogenesis, and discuss in detail the advanced bell stage of tooth development with the help of a neat diagram. You have to first write the contents, especially for essays. You have to write the contents, and then only you have to start with your essay questions. So the contents, like first you have to define odontogenesis, then you have to enumerate. You don't have to explain, but you just have to give the names of the stages of tooth development. Then you have to talk about the tooth germ and in advanced bell stage. So when we talk about tooth germ in advanced bell stage, first you have to talk about enamel organ and the components of the enamel organ, and which includes outer enamel epithelium, stellar reticulum, stratum intermedium, inner enamel epithelium. And so what you can do is you can explain the thing, explain these components like as it is in the early bell stage, and say what happens for that during the advanced bell stage. So that way, like enough explanations and enough content will be there in your essay question. Then you have to also talk about dental papilla and dental sac, and what are the various events that happen during the transformation from the early to the advanced bell stage, and what are the events that occurs during the advanced bell stage, and the starting of root development. So if you were able to explain up to here, I think you'll be able to score pretty good marks for in your essay. So if somebody asks you what is odontogenesis, it's very simple. It's the formation and development of tooth is known as odontogenesis. And here we are going to discuss about both the early and the advanced build stage, components of the tooth germ in early and its transformation to the advanced build stage. So if you talk about the stages of tooth development, we know that there are morphological stages of tooth development, which is what we commonly uses, and we have physiological stages of tooth development. So when an essay question is being asked, you have to write both, and you have to enumerate both. Enumerate means you don't have to explain each stage, you just have to give the names of it. So you have bud stage, cap stage, early build stage, and advanced build stage in morphological, and under physiological you have initiation, proliferation, morphodifferentiation, histodifferentiation, and apposition. Now next is we are going to see the parts of the enamel organ in the early build stage. So if you see here, this red arrow is pointing to the area where you have the inner enamel epithelium, and this yellow arrow in the other picture, which is a more closer view, is showing flattened cells just above the inner enamel epithelium, that is a stratum intermedium. And above that you have a brown arrow showing a network kind of cells, that is the stellar reticulum. And the outermost area you're going to have cuboidal cells in the enamel organ, that is the outer enamel epithelium. The blue arrows are pointing towards that. Okay, so these are the parts of the enamel organ, and we need to explain each of these parts of the enamel organ. So let's talk about the parts of the tooth germ in the early build stage. So you can divide them like this: enamel organ, under which comes the inner enamel epithelium, stratum intermedium, stellar reticulum, outer enamel epithelium. Then ectomesenchymal structures, which is the dental papillae and the dental follicle or the dental sac. So when you talk about inner enamel epithelium, the cells in the inner enamel epithelium, they are low columnar cells, and they are separated from the underlying dental papilla by means of a basement membrane. And this basement membrane is often thickened, and this is known as membrana performativa. Okay, and the way in which this inner enamel epithelium is arranged in relation to the dental papilla in the early build stage gives you an idea about what is the morphology or the blueprint for the morphology of the future crown. And later the inner enamel epithelium in the advanced build stage will transform into ameloblast and start secreting enamel. So this is what we can see, the inside invagination of the early build stage, and I'm just putting a blue outline for it, and see the mesial or distal aspect. And if you compare that with the mesial and distal aspect of an anterior teeth, you can nicely see that the morphology of the future crown is being decided during the early build stage. The way in which the inner enamel epithelial cells are arranged in relation to the over the basement membrane in relation to the underlying dental papilla. So that is what we said, like in early build stage, actually the way in which the inner enamel epithelial cells are arranged over the basement membrane gives a blueprint for the future morphology of the teeth. Now this inner enamel epithelial cells, they are columnar and the nucleus is at the center. But when they transform into an ameloblast during the advanced build stage, during the transformation to advanced build stage, they increases in its length and becomes tall columnar. And the length is around 40 micrometer with a width of around 4 to 5 micrometer. And the organelles, they start developing more organelles needed for protein synthesis, because they're going to secrete the enamel matrix. And the nucleus will shift from the apical or the proximal end, that is away from the basement membrane. That is known as reversal of polarity. This reversal of polarity is an important point, and you have to underline that. Okay. Now, so this is what we can see. The inner enamel epithelial cells, when they transform into an ameloblast, you can see the nucleus has been shifted away from the center to the proximal end, and the centrioles and Golgi apparatus shift down, and this change in the position of the nucleus and the other organelles are known as reversal of polarity. Now the next layer is the stratum intermedium, and it's a new layer of flattened epithelial cells of 2 to 3 cell layer thickness, which appears between the inner enamel epithelium and the stellar reticulum, and this is a layer that appears in the early build stage. I can see the yellow arrows pointing to the flattened layers of 2 to 3 cell layer thickness. So even though the number of cells are pretty low, these cells are very crucial for the enamel development, and they are rich in organelles and alkaline phosphatase enzyme content, and these are required for the differentiation of enamel epithelial cells to ameloblast and enamel secretion. In advanced build stage, they become a part of the reduced enamel epithelium. The next one is the stellar reticulum. They are star-shaped cells seen on the center of the enamel organ, and they secrete complex carbohydrates like glycosaminoglycans into the intercellular spaces. And the glycosaminoglycans are hydrophilic and will start absorbing water, pushing the cells apart. And so the cells are are forced apart, but they remain attached to one another by the desmosomes. So they take a star-shaped morphology. What is the function of stereoreticulum? One is mechanical protection. The main one is mechanical protection because it acts as a shock absorber, protecting the inner enamel epithelium and or the future ameloblast. And maybe it can also contribute a bit to the nutrition. So during advanced birth stage, this stellar reticulum will collapse and will become a part of the reduced enamel epithelium. We will come to that and in detail in the future slides. Now comes the outer enamel epithelium. These are the cells that line the outer part of the enamel organ, and they are cuboidal cells. They are not columnar, they are cuboidal cells, and they are separated from the dental follicle or the dental sac tissue, which is outside the enamel organ, by means of a basement membrane. And in advanced bell stage, again, they become a part of the reduced enamel epithelium. So when you're writing an essay, always mention about that particular layer of the enamel organ, and also mention what happens to that, what is the fate of that particular layer during the advanced bell stage, because the question is about advanced bell stage. Next comes the dental lamina. The dental lamina was the structure which had attached the enamel organ to the oral ectoderm, and by early bell stage they will disintegrate, and the remnants of this dental lamina which is remaining in the tissues are known as cell of Serre. And from bell stage onwards, enamel organ is not attached to the oral ectoderm, and especially in during early bell stage, the dental lamina begins to disintegrate and successional lamina is formed, especially for those teeth, deciduous teeth, where there is permanent teeth going to erupt after them. A successional lamella will be formed, and they are actually the lingual extensions of the dental lamina. You can see the arrow that is pointing to the successional laminae. When it comes to dental papillae becomes completely enclosed by the enamel organ. So these are ectomesenchymal areas which is enclosed within the invagination of the enamel organ, and the cells are closely packed there with fine collagen and capillaries. And during this stage, there will be an organizational influence of the internal epithelial cells, and the dental papilla cells adjacent to the internal epithelial cells will transform into columnar odontoblastic cells. And that is what happens during the transformation from the early to the advanced bell stage. And in the dental sac, it is actually the ectomesenchymal structure that surrounds the enamel organ and the dental papillae, and they are ectomesenchymal, and they are dense and fibrous, and from these structures, like later, cementum periodontal ligament and alveolar bone is being formed. Now we have finished now the components of all the tooth germ, and we have seen what is their function and how do they appear and what happens to them during the advanced bell stage. Now let us talk about what is a what happens during the transformation from the early to the late bell stage. So during the transformation from early to late bell stage, there is a lot of histodifferentiation is going to happen. Cells are going to undergo alterations, and that is because of reciprocal induction. So what do you mean by, and as a result of reciprocal induction, you see that the internal epithelial cells are transformed into ameloblast, and you get reversal of functional polarity. So what is reciprocal induction? That is the interdependence between ectodermal and ectomesenchymal tissues for their differentiation. So there is an interaction between the ectoderm and ectomesenchyme, and it produces tissue differentiation. That is what you mean by reciprocal induction. Let's see how the reciprocal induction happens during the transformation from the inner enamel epithelium to the for the early bell stage to the advanced bell stage. So what happens here is the inner enamel epithelial cells, which is above the basement membrane, and what you have beneath that is the ectomesenchymal cells, which is the dental papilla cells. And because of the organizational influence or the signaling factors that has been released from the inner enamel epithelial cells, this dental papilla cells will transform into odontoblast, a cuboidal or columnar odontoblast, and these odontoblasts will in turn interact with the overlying inner enamel epithelial cells and transforms them into pre-ameloblast, where the organelles have been changed, and the nucleus has been shifted away from the basement membrane. That is known as the reversal of polarity. And since the odontoblasts are the first form cells, they will start secreting dentine, and the dentine secretion by the odontoblast will stimulate the ameloblast, and they will start secreting enamel. They will completely transform into an ameloblast and they will start secreting enamel. And this is how the hard tissue formation starts. Once the hard tissue formation has started, we will no longer call it as early bud stage. We will start calling it as advanced bud stage. So the hard tissue formation is the turning point where we are going to consider whether it is in the early or the advanced bud stage. So once the hard tissue formation is started, because of the histodifferentiation, we will start calling it as advanced bud stage. So when you're writing an answer, you can actually write it in steps like this. That is, one, the inner enamel epithelial cells interact with the dental papilla cells and induce them to differentiate into odontoblasts. 2, odontoblasts interact with the inner enamel epithelial cells and transform them into pre-ameloblasts. 3, odontoblast secretes the first layer of dentine. 4, dentine formation induces the pre-ameloblast to mature into ameloblast and start secreting enamel. So if you can write like this as like numbered points, I think you will be able to, rather than writing it in a running way, I think you can impress the evaluator and you'll be able to score more marks. So this is what you can see, the early bud stage and the advanced bud stage. The difference between them is the hard tissue formation. Now in late or advanced bud stage, there are two events that is happening as a result of the hard tissue formation. One is the reversal of the nutrition source and the formation of the reduced enamel epithelium. So in advanced bud stage, after the formation of the hard tissues, the nutrition supply for the secretory ameloblast, usually they used to get the nutrition supply from the dental papilla, which is the closest source of nutrition for the inner enamel epithelial cells. You know that the enamel organ is ectodermally derived structure and they don't have any blood vessels in it. So they have to derive nutrition from the underlying dental papilla, which is a mesenchymal structure which has got blood vessels in it, and the nutrition will reach these cells by means of diffusion. Now what happens here when the hard tissue formation has happened, usually they used to get the nutrition, as you can see from the red arrow, from the dental papilla side. But since the hard tissue has been formed, this nutrition source from the dental papilla side is cut off. Now the next closest source of nutrition is the dental sac, which is again an ectomesenchymal structure. That is here, the blue arrow, here the yellow arrows. Now if you can see here, like there is a pretty large distance between the dental sac and the inner enamel or the ameloblastic cells. Like that space is because of the stellar reticulum and of the enamel organ. So you need to bring this nutrition source, that is the dental sac tissue, close to the inner enamel or the ameloblastic cells, and for that, so they are separated by ameloblasts with a large distance by stellar reticulum, as I mentioned before. So what happens is, in order to bring this dental sac tissue close to the ameloblast, the stellar reticulum will collapse, and the outer enamel epithelium, the collapsed stellar reticulum and the stratum intermedium forms a flattened layer of 4 to 5 cell layer thickness above the ameloblast, and this is known as reduced enamel epithelium. After the enamel formation is completed, the post-functional ameloblast will also become a part of this reduced enamel epithelium. Initially, they will be an active ameloblast, but once the enamel formation is completed, this post-functional ameloblast will also become a part of the reduced enamel epithelium. Now, and they will be clinging on to the crown of the unerupted teeth, and that is during the protective stage of the life cycle of ameloblast. Okay. So the advanced bud stage, you should also mention about the beginning of the root development. So here you know that you have the cervical loop that is being formed. That is, once the hard tissue in the advanced bud stage hard tissue formation has started, the outer enamel epithelium will loop inwards and will join with the inner enamel epithelium, and that is what you call as the cervical loop. As you can see the red like markings, and this cervical loop will proliferate downwards and will form into Hertwig root sheet epithelium, which is the primodia or the structure from where the root is going to be developed. So you need to mention about that also in the advanced bud stage. So here you can see the histology and where the Hertwig root sheet epithelium is. You can see the cervical loop beneath the hard tissues, and this the cervical loop proliferates downwards to form the Hertwig root sheet epithelium, and it is supported by the dental papilla cells inside and the dental sac cells outside. You can also mention about a few lines about the genes which are involved in the tooth development. For example, genes involved in tooth development are known as homeobox genes. The examples are limb homeobox genes, LHX6, LHX4, FGF8, SHH gene. Just have to remember and add these few lines, and it's going to give you more marks. It's just a trick for getting more marks. And areas of canines and incisors are controlled by MSX1 and MSX2. Areas where multicuspid teeth like premolars and molars, like you have DLX1 and DLX2. And areas where molar teeth are there, you have BARX1 gene along with DLX gene. It's very easy to remember. Just write that a few lines, and the evaluator will feel like, okay, you have studied a lot, and they will give you more marks. Okay. So that's all about the development of the the essay question. And now I hope all of you know like what you have to write for this essay question. Okay. So the next question that we are going, it is commonly asked and we're going to discuss in this particular session, is the enumerate the life cycle of ameloblast and discuss in detail amelogenesis. So you have to have an introduction. You can talk about what is amelogenesis in that. Then you have to enumerate the stages of the life cycle of ameloblast. You just have to name them. Now, then you have to talk about what is amelogenesis. Then you can talk about enamel secretion and enamel maturation. So these are the two parts that you have to mention in the amelogenesis part and the clinical considerations. Nowadays, like a few times they have asked to elaborate the life cycle of ameloblast. So if that a question is being asked like that, you have to mention about all the stages of ameloblast. But here in this particular question, you need to mention or enumerate the life cycle of ameloblast, or enumerate the stages in the or just name the stages in the life cycle of ameloblast and discuss in detail the enamel secretion and enamel maturation. So what do you mean by amelogenesis? It is the process of formation of enamel, and the ameloblasts are the cells which produces enamel, and it is differentiated from the inner enamel epithelial cells of the enamel organ. And the morphological and physiological changes that occurs in the ameloblast during its course is known as life cycle of ameloblast. And if you look at the life cycle of ameloblast, we can divide them broadly into three categories. One is pre-secretory stage, secretory stage, and post-secretory stage. In the pre-secretory stage, you have the morphogenic stage, that is for the inner enamel epithelial cells, and in the early bell stage, where the blueprint for the future morphology of the teeth is being determined, as we had been mentioned earlier. And then you have the organizing and the differentiating stage, that is where it undergoes reciprocal induction, and the inner enamel epithelial cells are transformed into ameloblast. And then comes the secretory stage, where, or the formative stage, where they are going to secrete the enamel matrix. Once the enamel matrix is being secreted, it is known as, the next stage is known as a post-secretory stage, under which comes the maturative stage, where the mineralization of this enamel matrix that is being secreted happens. Then once the enamel is being completed, the post-functional ameloblast becomes a part of the reduced enamel epithelium, and they will be covering the crown of the unerupted teeth and protecting them from the surrounding ectomesenchyme. That is what you know as the protective stage. And later, when they are about to erupt out, the reduced enamel epithelium and the post-functional ameloblast along with that has got a role in forming the eruptive canal and the eruption of the teeth outside into the oral cavity. That is what you call as the desmolytic stage. So you don't have to explain all these things if the question is being asked in this way. Just have to enumerate or give the names of these stages of the life cycle of ameloblast. And you can also draw a diagram for the life cycle of ameloblast. And you get the initial, the morphogenic stage, there's a locuboidal cell with the nucleus at the center. The organizing stage, there is a reversal of polarity. In the formative stage, you have the Tom's process that is being formed. In the maturity stage, you have rough-ended ameloblast and smooth-ended ameloblast. Then comes the protective and the desmolytic stage, where the post-functional ameloblast has got is again like a low columnar cell with the centrally placed nucleus. So now easily you can draw this diagram, not that complicated. So first let's talk about the enamel secretion. And at this stage, the ameloblast performs the function of secretion of enamel and partial mineralization. So there are three events that occur during this stage of enamel secretion. That is, first is the formation of junction complexes, second is the formation of Tom's process, and third is the secretion of enamel matrix. So let's go into each of these stages in detail. The secretory ameloblast will develop junctional complexes, And the reason why they have the cell-to-cell junctions, and the reason why they develop these junction complexes is, one, in order to maintain the integrity of the ameloblastic layer, and it also controls the metabolic diffusion, or like whatever has been secreted should not get washed away. So it maintains to make sure like the metabolic diffusion between the extracellular spaces has been controlled. So that is for what like the junction complexes are formed. And you have the initially what forms is the proximal junctions away from the basement membrane, and these junctions are usually leaky, they're not very intact. And then what forms is the distal junctions, and the distal junctions are pretty tight, and they will act as permeability barrier and prevents the calcium from reaching the surface through the extracellular space. So whatever has been secreted to the basement membrane area above the dentine will not get washed away. The distal tight junctional complexes make sure whatever is been secreted by the ameloblast will remain over there. Next is the formation of Tom's process. Now, the ameloblast will develop a distal extension, and that is known as the Tom's process. This Tom's process is important for the enamel secretion. The Tom's process is partially separated from the cell by an incomplete septa. See, this is how the Tom's process will appear. It is separated by an incomplete septa formed by microfilaments and tonofilaments. So this is called as the proximal part of the Tom's process. Then later, in the initial stages of enamel secretion, only proximal part of the Tom's process will be there. Okay, and they are separated from the rest of the ameloblast by tonofilaments and microfilaments. And after the initial layer of enamel is being secreted, a conical extension will develop from the on the Tom's process, that is known as the distal part of the Tom's process, like this. So both the proximal part and the distal part together is known as Tom's process. So the Tom's process, basically you have to remember that they are important for enamel matrix secretion. They are distal extensions of the ameloblast. There are two parts of the Tom's process. Initially what develops is the proximal part of the Tom's process, and later there is a conical extension that is known as the distal part of the Tom's process. So both proximal and the distal part of the Tom's process together forms the Tom's process. So let's talk about how the enamel matrix is being secreted. In the initial stages of ameloblast, they have a flat basal layer, and the Tom's process is going to be formed, but only the proximal part of the Tom's process will be formed in the initial few layers of enamel when they are secreted. And since the distal part of the Tom's process is also not there, the proper rod structure of the enamel will not be formed for the initial layers of enamel secretion. So you have the initial layers of enamel that has been secreted with only the contribution from the proximal part of the Tom's process, and so this initial layer of enamel is known as rodless enamel or a prismatic enamel. And after the initial layer of enamel is being secreted, the ameloblast migrates away from the dentine surface and they develop the distal part of the Tom's process. Okay, now let's see like how the proximal and the distal part of the Tom's process is going to secrete enamel. Now, the proximal part of the Tom's process is going to produce enamel known as interrod enamel, and the distal part of the Tom's process is going to produce enamel known as rod enamel. Okay, now let's see how it happens. So the proximal part of the Tom's process forms the interrod enamel like this. On around the proximal part, like the enamel matrix will be secreted, and this portion actually, and this enamel that has been secreted forms the interrod enamel like this. The enamel matrix will be secreted around the proximal part of the Tom's process in a cylindrical way. So what you get is because of the proximal part of the Tom's process, enamel matrix secretion, what you get is something known as a cylindrical kind of structure. Okay, because it is like around the proximal part of the Tom's process, the enamel matrix secretion is happening, and this is what you call as the enamel pit. So this kind of honeycomb structures you will see there. That is what you call as the enamel pit. So the walls of this enamel pit is formed by the interrod enamel secreted by the proximal part of the Tom's process. Now, as they are secreted, the ameloblast will move away from the secretory dentinal surface, and now what is there inside the enamel pit will be the distal part of the Tom's process. Now, the distal part of the Tom's process is going to secrete enamel and this enamel that is being secreted by the distal part of the Toms process will fill inside the enamel pit. Okay, and this forms the rod enamel. So here you can see the enamel pit will be filled in by the rod enamel secreted by the distal part of the Toms process. So now you can see, like, if you have multiple ameloblasts been put together, always the interrod enamel from the proximal part of the Toms process precedes the rod enamel secretion by the distal part of the Toms process. So initially what you have is the enamel pit, which is formed by the interrod enamel secreted by the proximal part of the Toms process, and into this area you have the distal part of the Toms process secreting the rod enamel. So this is what you can see, the electron microscopic picture. You can see the interrod enamel forming the enamel pit, longitudinal section, and inside the interrod enamel you have the rod enamel that is being placed inside. Now what you have to remember is that even though we call it as rod enamel and interrod enamel, both of them are same. It is only the difference between the only difference between the rod enamel and the interrod enamel is only in the orientation of their crystals. And you have to remember, as I told you before, the interrod enamel formation is always a step ahead of the rod enamel secretion. So as the enamel is being deposited and the rod enamel is being secreted by the distal part of the Toms process, the ameloblast will move away from the secretory surface. But as they are doing it, sometimes what happens is the Toms process, which is a soft tissue structure, is a cell, a part of the cell or a cell membrane, part of the cell. They get squeezed between the rod enamel and the interrod enamel, and that is what you call as the rod sheath. So here you can see the rod enamel and the interrod enamel. Okay, so I will show you here. So what you get outside is the interrod enamel, and what you get inside is the rod enamel. And you can see that interrod enamel and rod enamel is separated by an organic material that is a part of the distal part of the Toms process, that is known as the rod sheath. Okay. Now you have to remember that the distal part of the Toms process is not a cylindrical process. It is just a conical process. And that is the reason why, if you see here, the rod enamel is separated from the interrod enamel by the rod sheath in only 2/3 of the area. And in 1/3 of the area, you can see that the rod enamel is in continuation with that of the interrod enamel. And that is the reason why you have a keyhole shaped structure for the enamel rod. Okay, I hope all of you are clear about it. So later when enamel portion is being taken, we will talk about the enamel rod enamel structure. And you have to remember that this is because of this, where the rod sheath is separating the interrod enamel from that of the rod enamel by rod sheath, and only 1/3 of the area the rod enamel is continuous with the interrod enamel. Now remember, an enamel rod is comprised of rod enamel and interrod enamel. Okay, so don't confuse between these terminologies. So if you see the enamel rod, the rod and the interrod enamel together forms the enamel rod. Okay, and in transverse sections, the enamel rod have a keyhole shaped structure, and the head is formed by rod enamel, and the tail cervical to it is formed by the interrod enamel. And if you see here, each enamel rod is formed by 4 ameloblasts. That is, you can see that the head part is formed by the rod enamel of 1 ameloblast, and the tail part is formed by, or the interrod enamel part of the keyhole shaped structure is actually formed by the interrod enamel contribution from 3 other ameloblasts. So that is why we say that each enamel rod is formed by 4 ameloblasts, and each ameloblast contributes to the formation of 4 enamel rods. Now the bulk of the enamel is being formed now. Now in the last phases of the enamel secretion, again the distal part of the Toms process is lost, and we have only the proximal part of the Toms process, and they secrete with only the proximal part of the Toms process, they secrete a few layers of enamel. And since the distal part of the Toms process is not there, what we have is the rodless enamel or aprismatic enamel. So basically you can say that enamel layer is composed of rod containing prismatic enamel, which is sandwiched between rodless, aprismatic, initial and final layers. So when we talk about the mineralization of enamel, That is a little bit different from that of the other hard tissues. The other hard tissues, as soon as the enamel matrix layer has been secreted, undergoes mineralization. But in the case of enamel, the mineralization happens only after all the matrix has been secreted. The complete thickness of matrix, once they are secreted, only the mineralization process will start. And the ameloblasts are the cells which will perform this mineralization process. And when then this stage of ameloblasts, when they are in the mineralization process, they are known as the maturative phase. Okay, and at this stage, the ameloblasts will show a reduction in the height and volume and the organelle content. The mineral deposition of enamel matrix occurs in 4 phases. One is immediate partial mineralization, that is around 25 to 30% of mineralization of enamel will happen in the secretory stage itself. So we don't have to worry, it is not in this particular stage. Now the second stage, the mineralization will start from the surface and proceeds to the dendroenamel junction. And in the stage, the mineralization will rebound from the dendroenamel junction to the surface. And further deposition of minerals will happen on the surface alone. That is after the mineralization process is over. Now let's talk about how this really happens. The first is the immediate partial mineralization. As I told you, this is around 25 to 30%. It starts at the dendroenamel junction where the enamel proteins are present on the layer of dentine, and the initial crystals are formed by nucleation, and the initial crystallines elongates till it covers the entire thickness of the secreted matrix. Next is maturation, that is the gradual completion of mineralization. And here the ameloblast plays a vital role in or key role in this mineralization process. Now there are two events that are happening over there. One is there is a massive reabsorption of the matrix proteins. That is, already enamel matrix proteins are there, that has to be removed so that the mineral hydroxyapatite crystals can be deposited over there. So these proteins are broken down by proteolytic enzymes and they are reabsorbed by ameloblasts by endocytosis. And the deposition of the minerals will be happening after that, resulting in a rapid growth of mineral crystals. So these ameloblasts are the ones which are going to do this dual functions, that is removal of the protein and secretion of the hydroxyapatite crystals, that is secretion of the inorganic material and removal of the proteins and water to the matrix. And the ameloblasts which are performing these dual functions will show morphological alterations, that is that it can be smooth-ended ameloblast, which is actually removing the proteins and water, and ruffle-ended ameloblast, which is secreting the inorganic matrix. So the secretion of the inorganic matrix has been in maturation is run by the ruffle-ended ameloblast, and the removal of the proteins and water from the matrix is to provide space for the minerals is done by the smooth-ended ameloblast. So here you can see there is the enamel, that is a yellow block over there, and the ameloblast is there. And I'm just showing you that you have the ruffle-ended ameloblast, which is actually secreting the enamel, the hydroxyapatite crystals. Then you have the smooth-ended ameloblast, which is actually going to remove the proteins and the water and the organic material from the ameloblast. So this dual function will keep happening. Okay, so the ameloblast will be smooth-ended one time and will be removing the protein and the water, and then it will be a ruffle-ended ameloblast and will start secreting the hydroxyapatite crystals. So again, it becomes a ruffle-ended ameloblast and it starts secreting the enamel matrix. Then it transforms again into a smooth-ended ameloblast. Okay, so like that. When it is a ruffle-ended ameloblast, and that is a time when it is actually secreting the hydroxyapatite crystals, the proximal junctions are leaky and the distal junctions will be tight. The proximal junctions you can see are leaky and the distal junctions are tight because whatever hydroxyapatite crystals are being secreted, that should not get washed away. That is why the distal junctions will be tight. The red arrows are pointing to the distal junctions which are tight, and the green arrows are pointing to the proximal junctions which are leaky. But when it comes to a smooth-ended ameloblast, whose whole purpose is about reabsorption, you have the opposite thing that is happening. You have the distal part of the junctions which are leaky, because you will have more absorptive areas and for removing the water and the proteins, and the proximal part will be junctions will be tight. So this phenomena where in which the ameloblast changes its shape or morphology and performs dual physiological function is known as modulation. So the definition of modulation is cyclic creation, loss and recreation of a highly invaginated ruffle-ended apical surface or a smooth-ended apical surface is known as modulation. So you have to remember the definition of modulation. You have to underline that. With this, I think the matrix secretion and the maturation is over. Now you can also add a small note on the clinical considerations, and we can talk about amelogenesis imperfecta, something that you're going to learn in third year. But still, if you can just mention about a few lines about it, it's going to give you more marks. You have three types of amelogenesis imperfecta: hypoplastic type, where the defect is in the enamel matrix secretion, and due to the disturbance in the differentiation of viability of amnoblast. Hypocalcified, where the defect is in the calcification. Hypomaturation, it is due to the alteration in the rod structures. Okay. So with this, this portion is over. So thank you so much for your patient listening, and best of luck for your upcoming exams. If you have any queries, you can put it in the chat box.